Hello, hello.
Oh, we didn't even talk about that. Okay, tomorrow major defects. In fact, you do it by open chromatin. So it's just sequencing has a lot of power. Whatever you attach to it, makes it interesting, right? Anywhere? Okay, we'll cover the methylation sequencing tomorrow. All right, so the less accessible region will look like this, and more accessible region will look like this. So wherever this is, there will be no peak. There's an open chromatin, there'll be a peak. Open chromatin, peak, open chromatin, peak. Very simple, okay? This is a one-step process. This is a very simple one-step reaction, and that's why everybody's adopted it, because it's easy peasy, right? You take a reaction, put it in your thing, it makes the adapter, you isolate it, and you sequence it. Very simple. Okay? So ATAC-seq had taken the world by storm at some point. Okay, 3D chromatin organization. I will talk about ATAC-seq tomorrow, and also I want you guys to take you to the step. The concept is the same. Okay, 3D chromatin organization. This is the interface nucleus. If we focus on one region, you can see something like this. Okay, this is called topologically associated domain, TAD domains. That's fundamental, one of the fundamental things to 3D chromatin organization. I told you guys earlier, looping mechanisms are key or the basic unit of 3D chromatin organization. Once you loop, so within a TAD domain, can you see multiple loops? Sort of, somewhere. It appears that TAD domains have many loops like this, what you see, okay? So it is made of many loops together. The next layer is called TAD domain. Loops is the lowest unit. And what happens within this? Two regions come together, A and B here, intrachromosomal interaction. They can be mega bases away, kilo bases away, many bases away, okay? That's how we talked about enhancers doing it, right? Then there are interactions between two different major domains, right? Something like what is shown on the thing. So that's a next interchromosomal interaction. So basically, yellow was chromosome 1, blue was chromosome 2. There are regions between two chromosomes that interact in chromosomal in the interface, okay? So you could have regions that are in chromosome 1 regulate the region genes that are in chromosome 2. It happens all the time. Okay? It doesn't happen as often as, of course, the regions that are in proximity. It happens. So linear conformation was that. The 3D loop conformation is that. So now it brings the red element close to the blue element, right? Red element was could be an enhancer, could be an insulator, could be X, Y, Z, anything. It just brings two things together. So what was bound to red now can access blue, okay? Or what was bound to blue can access the red. So that allows two regions to interact together, regulatory proteins that bind in the two regions to regulate the genes far apart, okay? And their gene goes up, gene goes down. How do you identify this? This is called Hi-C, a technology called Hi-C, which is what I was telling you earlier. Eric Lender published his first paper in 2009 with a technology called Hi-C. So we were able to get genome-wide glimpse into how 3D chromatin organization at the genome-wide level. Okay? How does this work? Okay. how it works is, simplistically, it's a little complicated. So I'm trying to think about how to explain this to you guys easier. I do have another slide. I can put up another presentation where I can explain high C a little better, probably. But think about this, what we've got here, right? A loop, a bowl, noodle in a bowl, okay? We do something called cross-linking, just like everywhere else, right? Cross-linking brings the two things that are in proximity together through a formalin, CHO bond, right? Okay, formaldehyde bonds, essentially. So these two regions can come together. That's what is showing by black, okay? Now what I do is I use a restriction enzyme that frequently cuts in the genome. A restriction enzyme. What is a restriction enzyme? Known sequences. A restriction enzyme that will cut at a 6 base pair region will be less frequent in the genome than a restriction enzyme that cuts with a 4 bases, right? Did I make the right statement or wrong statement? Right? Because the frequency of a 4 base pair restriction site is larger than the 6 base pair restriction site. So we use a restriction enzyme that cuts 4 bases so that we can cut frequently, okay? An enzyme that is used frequently on this is called Hindi3, and there are others, okay? Basically, so you cut it, so you get these fragments. You get this? Hindi3 is probably not a 4 base pair cutter, by the way. So there might be a, but nonetheless, you cut it with an enzyme to make it like things look like this, which you're looking, which is good. You got this, right? You guys got it? Then, I think he's made it wrong, to be honest. But you fill it with something called DC, a biotin labeled DCTP. So you do a filling reaction in the end by putting something called biotin labeled cytosine, CTP, okay? So basically biotin is what? You guys know what is biotin? Biotin binds to beads called streptavidin beads. There's a very strong reaction between streptavidin and biotin. If you don't know this, go look it up, okay? So you can fill up the region with a biotin labeled DCTP. You fill that up and then you do a ligation under dilution condition. So only the regions that are ligated, that are next to each other, gets diluted, okay? Maybe I need to do a better explanation of that. Dilution under ligation, ligation under dilution conditions. So ligation under dilution conditions will only happen if the two regions are in close proximity. That was the key to this. People figured out that if you, let's say, normally we do these reactions in like 10 microliter, 20 microliter, but if you do it in a 1 ml reaction, okay, then you're going to only preferentially ligate things that are next to each other. That was the fundamental to this. So you do ligation under that kind of condition. So you ligate, you now take out everything else, and you got a DCTP, remember? So you can pull down that region that has biotin tag filled in, right? And you get these regions, and then you go in and sequence it, all right? So what were we looking for? We were looking for things that are in close proximity. That was the objective in this method. Things that are next to each other, and not do those that are not next to each other. So you did two things. One, you cross-linked first, and then you did the ligation of the DCTP, that reaction under dilution conditions, okay? And you got this, you filled the dot DCTP ligation, I'm sorry, the ligation of two fragments, okay? So two fragments get ligated with each other. That's what he's trying to show, okay? So when you now sequence from one end versus another end, one end maps to a region which is here, okay? And the other end breaks to the region that is on the other end. So now by mapping that, and saying, okay, this was in this position and this was in this position, now I know these two positions actually interacted with each other, right? So you created a fusion transcript by ligation and a dilution condition. Does that make sense? Okay, so this ligation here happened between these two, right? And he created these. So now you filter out reads that are just looking to one. You don't take them. You take only the reads that are fusion, that are in two different spots in the genome. And that's how you get it. Once you do that, the map becomes something like this, a triangular pyramidal map. Do you understand any of that? No. What does that tell you is the reads. I'm trying to see how to explain this better. Okay, so when you map these sequences, right, what you go see is something called interaction frequency. Interaction frequency of will be of this region to this region here, and you essentially are plotting on the y-axis. So this is usually a square. Let me actually let me do it tomorrow. I think I have better slides than this. But this is contact maps, and this essentially what this means is that wherever the triangle ended, there are a lot of interactions within that triangle. That's what it means. Okay, I'll do a better job tomorrow. I need to figure out. I have a slide deck somewhere on this. So basically, in this region and this region, these two regions interact at frequencies here. So anything between this region, interaction frequencies here. Okay, but this region does not interact here because there is no contact. Okay, this region interacts within this domain here. This is called a TED domain. Those triangles are TED domains because within those TED domains there are a lot of interactions. Remember, we looked at that, right? Like, not a great job. I'm sorry. Need to do better. Here. So you see how many interactions are there, right? So you will see a lot more interaction within a certain region. So that triangle essentially represents this. So there are lots of loops within that. So each of those dots, red dots in that is actually a loop. Get it? Okay. And these, whatever you see here, these looping, there are many of those inside this, right? So you're going to get a lot of such interactions in the triangle. You did not understand that. Okay, next time, let me do a better job next time, okay? But basically, what you can try to remember for your level right now is to say this triangle, there are a lot of interactions. Anything that is deep red is essentially contact frequency. How frequently those two regions will contact with each other. That's what it is. So it is TED domain here, TED domain here, and there is much smaller number of interactions because that's all. So this is the contact frequency. You see this, low to high, right? So there is low interactions here, there are higher number of interactions here. And the whole genome is like this. So within that domain, that's it. As I said, there are multiple interactions within that. I'll explain this better next time, okay? Let me get my slide deck. I think I do have a better slide deck. This is how it looks normally. So this is represented here in a 6 MB resolution. Okay, again, it's not going to be easy because this is a tough method to actually explain. This is how, within that domain, if you go deeper into this, this is how it looks. You see this checkered pattern? This pattern is within each of these. This is now here. Okay. That's at, and then within each of these is these. So at a lower resolution, you can see these contexts that show you these loops. So you see the loops here, right? These loops are here. These loops, when they assemble over each other like this, then they are here. So they are interacting at different places. So anything where there is a red means there is an interaction, right? High, deep red. Where there's a light red, there is not as much an interaction. Okay. And then when these make bigger, these are called chromosome territory. So we have these, within these we have these, and within these we have these. Okay. So many of these make this, and many of these will make this. And our whole chromosomes are in compartments. In compartment A, compartment B. So that's what it's showing on this side. Okay. Compartment A is euchromatin, compartment B is equivalent to heterochromatin. So in compartment A, there is always positive interaction, like there is open interactions, and then compartment B there is heterochromatic regions. Very simple. It always tracks with that. Okay, so that's a bigger compartment size. We saw that, right? I mean, in electron microscope, you see dark places and you see not so dark places. Not so dark are euchromatin. Those euchromatin will actually make a separate sort of structure, and heterochromatin is a separate sort of structure, which makes sense. Does it? No, right? Sure. Now within heterochromatin, when you go in, you see more deeper interactions like this, and in euchromatin, you see interactions like this. As I said, I'll try to do a better job of Hi-C. Hi-C is the key technology that actually has discovered all the 3D chromatin structures. So I think we need to better understand it. It is not easy. It was never easy for me to understand when it came out. So it takes its time. I think it's not everybody. I mean, not people like me are not very good at explaining this. Let's just put it that way. And it is dynamic. So at one time it will be this, and another time it will be this. It's actually not entirely true. A lot of, these chromatin interactions tend to be very stable normally, but in some places it will be dynamic. Okay, and when it's dynamic, it's going to change the interaction levels, which you can see, of course, ABCD, ABCD is trying to show that, and XY, XY, so they can be together, they can be off. And that's one of the ways you can regulate, chromatin interaction, and then eventually gene expression. Okay, so that's a summary, but let me go back and figure out the better way of Hi-C. But just to get you guys to think, those triangles are domains, right? And many of those triangles into higher order is compartments. Within those triangles are loops. The loops are anchored together by CTCF and cohesin. We looked at the CTCF, and CTCF is the one who actually also organizes these 3D chromatin. PRC1 that we studied yesterday, polycomb group 1, plays a very important role in these 3D higher order compaction, and then histone H1, etc. that we've looked at, right? So basically, these are the regulators, and this is how the structure is, but I'll do a better job of Hi-C tomorrow. I'll figure out my deck where it is, okay? And then go from there. Okay, any questions? I'm sorry, this was not perfect, I know. Any questions? We need to get out. This was confusing to most of you, right? Huh? I guess. But you guys get this part. You get the part that there is higher order structure, then you get this part, there's compartments within that, you get this part, there are loops within that. Well, that's all matters for now. You don't have to worry about the method for now. Method we'll worry about later, okay? Methods, very few people can actually do very good Hi-C. I have one guy in my lab who only does really good Hi-C. Otherwise, it's a very difficult technique to master. And it does not happen. So we spent millions of dollars doing this, and you don't get good data. But that dilution, ligation or dilution condition is very difficult to optimize. But when that happens, you see this. So there are only few labs actually who can do very good Hi-C and very good data set generation after this. Okay? I would like to at some point teach you guys single cell experiments, because all you're learning is bulk assays, and we do tons of single cell RNA-seq, single cell ATAC-seq, etc. But you guys are too naive for this. That gets much more complicated. So single cell, this is all going to the single cell level. But if you want to understand it at one cell, how is this happen? In things like cancers or other tissues, every cell might be slightly different. And there are groups of cells that behave the same, and there are other groups that don't behave the same. So biology has that layer of complication as well at some point. So a lot of research in last 5-7 years, the most cutting edge research currently that's happening has happened in single cell space, and now it's happening in the spatial level, where we know in tissue where things are happening. How is the organization at the spatial level? So all of these technologies that I'm telling you are actually quite old. 10 years old in the real world of keeping up with the best of the best. Okay, now everything has moved into single cell space. Okay, does that make sense? No, single cell. If you guys, those of you who are PhDs and you want to do postdocs, you will have to think about, the latest and the greatest, and the latest and the greatest are that kind of technologies. Okay, so when you go on to choose a postdoc position, if they will take you, of course, there is money. So yeah, tomorrow I'm going to try to get to DNA methylation mapping, which I have the slides for a little bit, which is very easy. since we've done everything else. I'm going to try to give you chromatin remodeling, chromatin remodelers. We're going to have to finish up on high C if I can find a better slide deck. If I can't, then you can contact me. I'll explain to you later. Okay, but if I can, then that would be great. What else do I need to teach you guys? There's some disease stuff, but there will be somebody else will be covering it. I have some of that level that I can teach you. And then I had to teach you neuroepigenetics on Friday, which I'm not going to be able to teach you. I think I'm going to have to do slightly different stuff. Because then I think I would like to teach you guys transcription factors and how they are master regulators and in cell differentiation, because then that will complete the whole story in your mind. Other neuroepigenetics you can read later on. I mean, I'll try to touch on it in one of the sections, but I think probably more important is to know the hierarchy of transcription factor circuits and how they then use chromatin to really drive the cell fate. And we'll be talking about iPSC, induced pluripotent system cells. Have you heard of that? iPSCs. I think I want to teach you guys that. That's probably more important, than teaching you neuroepigenetics. I'll touch on that. I'll touch on neuroepigenetics, but iPSCs is more important because the transcription factor hierarchy becomes very open and appear there. And also there's a lot of role of all this that you've learned so far. So what should be, at the end of the day, in my mind, what should be clear to you is how this whole thing works in cell differentiation and as such, little bit in cancer. And then somebody else will teach you cancer epigenetics, I think on Sunday. Okay? Is that cool? Sounds good? Okay. Anything else? And we need to still dance tomorrow or day after tomorrow. What do you guys want to do tomorrow? No? No team building exercise? You're like, enough of it. Because otherwise I see your faces and I'm like, sad. At least I can make you happy in the morning. Right? Today was fun. What else would you want to do? No? Nobody knows what they want to do? Anybody else? Group activity? Sure, but you guys have done that. Let's do something else. There are more things to do. You guys want to solve some puzzles together? Yeah? It'll be fun? Okay. We'll try something that tomorrow. That's again another team building exercise. Or we can do some pranayam together, right? Huh? Chalega? You are the fan of what is his name? Ramdev? No? Come on, he's changed the lives of so many people. And what a great man, has made a lot of money, but has actually brought yoga to everybody's household, right? I admire him for that. It's amazing that he's actually able to get people to do Kapalbhati or pranayam. How many of you really took pride on it before that? Oh, what is this? Right? Okay. What else did I want to say? Guess I'm waiting for Santosh to come back because we have a meeting with your Vice Chancellor. Let me see. Are you guys tired? No, I'm sure. Okay. Oh, I was asking for feedback yesterday. Did anybody think of any more feedback? No? Come on, guys. No more feedback? No feedback? Nobody wants to tell me how bad it was? You can tell me how good it was too. I'll take that. What's good? What's bad? Oh, actually, I did have this to teach you guys. Oh, man. All of this. I need to get to this tomorrow. Okay, maybe we'll start with this, all of this tomorrow. You guys want to know about diseases? We just talked about 5-methyl-C, right? That's all. We didn't talk about bisulfite. Oh, yeah, all of this stuff. I'm sorry. I ended there. Yeah, there's a lot of stuff here to teach you, actually. Not a lot. I mean, it's fun. Methylation mapping. This is all methylation mapping. Oh, yeah. Okay. Let's see if you have time tomorrow. Maybe go do that and then do the chromatin remodeling. Yeah, tell me. Feedback. You need forms. You can write it in the notebooks you guys have and tell me. I won't know whose handwriting is what. No? Huh? That's a good idea, right? All right, why don't you guys write that? Bring that to me tomorrow morning. And I won't know who's what, right? But I would like to know how bad am I doing or how good am I doing. Because my goal is to take the good things forward and improve on the bad things. All right, have fun, guys.